Alright guys, welcome back to Underground Science, and in this video we're going to be talking about mismatch repair. So this is a single-stranded repair mechanism. Okay, so let's go and see how this works. So why do we do mismatch repair, right? So you know how DNA polymerase, we said that it had a 3' to 5' exonuclease activity. That's the proofreading activity of DNA polymerase. But what if it that doesn't work, right? So let's say it does proofread some, but let's say there's one or two that DNA polymerase put down the wrong base and it couldn't proofread and correct it. So that's where mismatch repair would come in. Okay, so let's see how this works. Specifically, we have mut S and mut L here, but we should focus on more. Okay, so mut S is actually a complex. Okay, it's actually a mut S alpha complex. Okay, and then same with mut L, it's a mut L alpha complex. Why do I say complex? Because these are made up of proteins. Okay, so mut S alpha is made up of MSH2 protein and the MSH6 protein. Okay, mut L alpha is made up of PMS2 protein and the MLH1 protein. Okay, so PMS2 and MLH1. So that's what we have to know for now, because it's going to come into play later when we talk about Lynch syndrome, okay, which is relating to a defect in mismatch repair. All right. Okay, so let's see how this works, right? So first what happens is mut S alpha will come to the site where we have to repair, okay? Because notice this is cytosine and this is not guanine. It's supposed to be guanine, but it's not, it's thymine. So we got to take this whole nucleotide out, so the sugar phosphate backbone plus the base, and replace it with the correct nucleotide, or correct base, which is guanine, right? So mut S alpha will come here, wherever we have to replace, okay? And then it's going to recruit mut L alpha, okay? Mut S alpha will help recruit mut L alpha, which will come to this place again, and it's going to make a nick, okay, like a small little break, okay, in the sugar phosphate backbone. And it's going to do it in this case, we'll talk about on the 5' end, okay, in this case, because that's what our example is as we go more down. So let's say mut L alpha comes in and makes a nick, right, near this side, okay? So this side is the 5' end, and then this side is a 3' end. Mut L alpha will come in and make a nick on the 5' end, okay? That's going to give exonuclease 1 the opportunity to bind near the nick and start taking out the bases, or the nucleotides, okay? So let's say exonuclease 1 comes in and binds here, okay, near the nick that was made around here by mut L alpha, okay? So mut L alpha, let's say, makes the nick here, so then the exonuclease 1 can come in and bind near the nick and then cleave out nucleotides starting from the 5' end and going towards the 3' end, okay? So mut L alpha makes the nick, then exonuclease comes in and binds near the nick and starts removing the nucleotides from the 5' end towards the 3' end. Okay, so it's going to remove a whole chunk, not just this nucleotide that's wrong, okay? It's going to remove a whole chunk. So let's see how that's going to look like. So that in this case we see the cytosine, the guanine, the thymine, and the cytosine got removed, right? And plus the sugar phosphate backbone got removed. So the next step is to replace these with the correct, replace these nucleotides, I should say, with the correct nucleotides, okay? That's complement to this strand. So what's going to happen is we're going to use this strand as a template in order to make in order to put correct bases here on this strand. So DNA polymerase is going to do this, okay? So DNA polymerase will come in and put the correct nucleotides in, okay? So what are the correct nucleotides in this case? Let's go to our normal colors. So for guanine, we're going to have cytosine. For cytosine, so this one was the wrong one. It's going to replace it with the correct base, which is guanine. Then this is going to be guanine, and then this is going to be cytosine, right? So now we have all correct complement bases. Okay, so now it's all cool. But then DNA ligase can come in and seal any other gaps that there are, okay? Because if there's any other nicks or gaps, DNA ligase is good at sealing that in together. Okay, so let's say after DNA polymerase put the correct bases, right? Let's say there's small nicks or gaps between these bases. like DNA ligase can come in and seal the sugar phosphate backbone together so that there's no breaks, so it makes it better. All right, and I mean, that's basically it, right? So that's mismatch repair, because now we have all correct nucleotides. All right, so this is really important when we're going to talk about Lynch syndrome in the next video. Okay, so yeah, that's basically it, and I hope to see you guys in my next video. Thanks for watching.